The final part of genetics would consist of translation. Translation is a process where this mRNA strand which we formed, is going to be converted into different proteins. So how this is going to happen is, first of all, this mRNAs are template, okay, and there's a different type of RNA known as tRNA, which is denoted over here, okay, here. So the tRNA, what it does is, it is your reading mechanism and it attaches a specific amino acid to the protein chain. So this is your mRNA and there are going to be codons or triplets over here, which is going to be read by this tRNA, and each tRNA has a specific anticodon which is its unique identity, and each anticodon will code for a specific amino acid. So there are 21 amino acids in total. So what happens is each of these has a code. Like for example, you see this GAC and CAG. All these are codes in the codon which correspond to specific uh, amino acids. This is known as the genetic code, and this is your base for protein formation. So first of all, before this tRNA goes on to read the mRNA, it has to be loaded with amino acid. That process is known as amino acylation of tRNA, or charging of tRNA with an amino acid. So the starting amino acid for any protein is methionine, and there's a specific starting codon for that. So what happens is, as if you see, this would be the mRNA and there are 50S and 30S ribosomes which are attached over here, and this tRNA goes and binds to these ribosomes and a protein is being attached each time, and so it's basically like going to keep stamping on proteins together, and you're going to get a protein amino acids together, and you're going to get a protein structure like this. So this is the first step of protein formation. Please note, only the primary structure, that is the order in which the amino acids are arranged, are done in this process, and it is all based on the codons present in the mRNA with the help of tRNA. So this would be the essence of uh, translation, and again this is further proofread, and there are many genetic error correcting mechanisms, or if if our cell detects any change in the genetic code or codons, or there is suddenly thymine present instead of uracil, it knows how to remove that part and correct it or degrade that part of the DNA and repair it. These are known as DNA repair mechanisms. So you just know that there are a few mechanisms to take care of all of this, and this would give you a very good idea of how our we grow as a organism. Everything is based on this. So that would be all. Thank you. Please do like, subscribe and comment.